Patrick. I'm a scientific software developer in Duca Hextra's lab, and I'm the primary developer between Laue-DIALS. So Laue-DIALS is intended to be an open source free replacement uh, for other software suites like Precognition is one many of you are familiar with, or things like the Daresbury-Lowy suite. So many of you are probably familiar with this, given we're at a biocar seminar, but the advantages of Laue X-ray diffraction, one of these are photon fluxes, short pulses, especially if you're at XFELs, which are great for time-resolved studies. And probably the biggest benefit, or at least one of the biggest benefits in my mind, is that you get full reflection intensities on a single image. So encapsulated within the Ewald spheres here, for the different wavelengths, you get a full reflection as opposed to a slice of a reflection that you get in monochromatic stills. So for anything except for these very low resolution spots, the partiality problem that is common in crystallography is basically completely eliminated by getting full reflections on single images. So currently used software, we have Precognition, we have the Daresbury Laboratory, Laue software suite, and then there's also individual algorithms that are often put into other larger software libraries. So an example of this would be Pink Indexer that is in an old version of the CRISPHEL suite for XFEL serial data. And this is more or less what most people work with. This and custom scripts that they write these days. This is how people analyze Laue data. But we would like to have a modern, open source, and free software that people can use, ideally one that's maintained well and extensible. And so that has been kind of the motivation behind building the Laue-DIALS software. It's free, it's open source, it's modular and extensible, and as the name hints, it's based on the DIALS package, which is in the CCTBX ecosystem. A lot of people have used this package for analyzing monochromatic data in the past, and it's now being extended in various directions. In my case, in polychromatic Laue-DIALS. I know others are extending it more for electron diffraction and for neutron diffraction as well. So DIALS is kind of our core dependency. And one of the nice things is it uses modern Python and is migrating its systems to more modern packages like using NumPy and SciPy and such. In addition to that, so we're not working with older technologies. So first, I'll give a couple of illustrations just of data sets we've been able to successfully analyze using Laue-DIALS. Some of these will be also shown in the paper. So these few examples I'm going to show just for versioning context uses DIALS 3.17 and Laue-DIALS version 0.4. That is the version that is in the paper and in the data depositions associated with the paper. So you can reproduce these using that if you like. Currently, DIALS, I think, is on version 3.22. So we're working on updating compatibility with that. But first here is this hen egg white lysozyme. And this is a relatively large data set. So it was about 3,049 images. And in particular, what we're interested in from seeing this data set is anomalous scattering from the iodine and sulfur molecules or atoms that are in here. And we get pretty good results. So these are the CC half values and the CC anomalous values we get in the data set. Pretty decent. And looking specifically at anomalous peak heights, we can extract them successfully from the data set using Laue-DIALS processing in conjunction with Careless and Phoenix. Another example is this PDZ2. This is an example of a time-resolved EFX experiment. So there are 4 passes at these various time points off 50, 100, and 200 nanoseconds. And then they also vary a little bit with their starting angle and their angular spacing. So they might be 1 degree, 2 degree, 4 degrees apart between these passes. And we can resolve difference maps between these. So on the left here, and you see these difference maps that we're able to produce between the passes at the various time points, and then also for electric field on and off, you can see these orange and purple arrows and the densities associated with that. And so we're able to resolve residue difference maps in EFX experiments and time-resolved experiments also, which is great. And here the CC sim of the PDZ2 EFX data, you can clearly see a difference in the trend lines for each of the different time points here. So we're able, that's exactly what you should see for an EFX experiment. We also tested on simulated data. So Derek was very helpful here. He helped us to simulate data based on the DHFR molecule, and we simulated many sets of data. How we decided to do it was to use a reference data set that had a certain set of parameters. So this would have been like a divergence of, I think, 0.1 degree, a mosicity of maybe 0.1 degree, and then the biochar spectrum, which is roughly 5.5% bandwidth. And then we simulated many data sets that differed from that reference data set in one key parameter. And so here are just some close-ups of reflections for each of those data sets. So we changed the mosicity for one scan, we changed the bandwidth for one scan. And to be clear, these bandwidth are simulated using Gaussian spectra centered on 1.04 angstroms, I think, or thereabouts, the center of the biochar spectrum. And then just applying a bandwidth for the Gaussian spectrum. And so this is just an example of what those reflections looked like on the detector. And we did this and we investigated how well we could assign wavelengths because it's simulated data. So we were able to know exactly what the wavelengths were that the reflection was simulated at. And so we tested the reflections, our Laue-DIALS code, the wavelength we assigned them compared to the true wavelength it was simulated at. And generally speaking, except for highly mosaic crystals, we achieved pretty good results. And even for highly mosaic crystals, most of the density is still on that linear line, although you get considerable off-axis density now also. We also have some merging CC halves for these the same data sets. You'll note that the high mosicity data sets don't go to nearly as high resolution, which is something you might expect. The dashed line that you see on these represents the reference data set. So that's the data set that the others differ by only this parameter from. And looking at also the completeness, interestingly enough, you'd expect it to go down a little bit maybe with mosicity, but interestingly enough, with our data, we get better completeness with higher bandwidth, which is not a huge surprise because that's one of the major perks of Laue techniques in general, is that you get this large envelope that you can encapsulate full reflections in. And so you need less data, which means the same amount of data gets you better completeness. So now I'll talk a little bit about how you use Laue-DIALS, what's involved in actually using it. This is a standard pipeline for what we call scan data, or you might refer to rotation series, a single crystal data. Essentially, you put a crystal on a goniometer, leave it still, take a snapshot, rotate it a degree or 2 degrees or whatever your spacing is, and then do that again. I say this just to emphasize we're not rotating the crystal through the beam as we're taking the snapshot. It's a still, but their stills are all correlated in terms of the crystal orientation. So this is our standard pipeline for that. And we have a command line interface right now. So we have documentation for this online. And these are right now the commands that exist in Laue-DIALS so that you could use these in a terminal without having to write any Python scripting of any kind. And this will do the functions for it. And so we'll just go through just a quick example of a DHFR analysis from real DHFR data that we have on hand. It's about 180 images, I think just slightly less, with 1 degree spacings. So the first thing you'll do is an import command, and this comes directly from DIALS. DIALS has a lot of inbuilt technology for importing data from different types of file types and headers, so CIF data or MCCD data, and it has useful options that you can use to override the data that's in the headers if that data is known to be wrong or not completely accurate for any reason. So in our case, these were MCCD data that we collected at Biocars. And you'll see that some of these options that I provided are, so for example, the scan oscillation start at angle equals 0, 1 degree spacing between the images. assigning the beam a wavelength of 1.04 angstroms, a pixels.mask file, which you can make, and I'll talk about in a little bit. And this is how you would import the data. And this will be true for anything you analyze in dials or any derivative packages. And so we use the same thing in Laue-DIALS. And then we use spot finding. So a few of these programs that are in Laue-DIALS are essentially rather thin wrappers around dials functions of the same name. So there's a dials.find spots. In our case, we use a Laue.find spots. And mostly what these wrappers do is set a few options in the dials version of it that are pretty commonly universally used for our Laue purpose case. So for example, in spot finding, one of these that you won't see here because it's set by default is using a 2D spot finder as opposed to a 3D spot finder, because all the centroid data should be on the image. And in particular for stills, we'll want to 2D spot find. Was there a question? No? Okay. So that for spot finding, I would note that the gain is probably the most finicky parameter, the gain that you set your detector at. And this is more or less a setting on how many spots you want to find on the detector. So setting this higher or lower will get you more of the spots or fewer of the spots. Setting it too far in one direction will either get you spurious spots, or you'll see very obviously that there are spots on the detector that you haven't actually captured with this spot finding tool. So I usually like to iterate this with using the dials image viewer, which will show you the image with the spots overlaid on it like this. So the black is the raw data reflections, and then the red are the spots that we're able to find. And you usually want to adjust this gain parameter until you roughly have something like this, where you can find all the spots that you could visibly see on the detector. Another useful thing of the dials.image viewer is that you can create a mask for areas of the detector that are clearly not areas that you want to find reflections in. So you'll see here in the bottom left, we have this giant black blur. That was from collimator scatter in this particular experiment. And so that's something you'd want to mask out. So we could build one with the dials.image viewer, and then you can mask out those spots. There's also tooling for masking out like the borders of images. So you'll see that on the far right here. There's a border mask there too, if you have any weird effects of the borders. And then this mask file will carry through the rest of the pipeline in the metadata for the experiment files that are in there. So you don't need to worry too much about providing it again. Laue.index is again a thin wrapper around dials.index, and this essentially in this version of Laue-DIALS and for scan data generally speaking uses the FFT 3D algorithm that's in dials, and that is inherently a monochromatic algorithm. We use it here for providing an initial estimate of the unit cell and the orientation of the crystal. And one of the things that's nice also about this is if it's scan data, there's also scan varying refinement that's available to the index data. So you can vary the parameters of your crystal over the course of the scan. So different images can, slightly adjust the ABC axes as needed, or other parts of the experiment, of course, you can do that as well for. And so we find often that the percent indexing success for this out of the reflections is often somewhere between 10 to 25%. I expect this would probably change depending on exactly what your beam spectrum looks like. These are from BioCars data. And having looked at multiple data sets, it's almost always between 10 to 25% for this monochromatic indexer. But that's not something you should worry about too much, because if it's scan data, we split it into stills at this point. If it's stills data, you don't really need to do this step. And then from there, we have this program called Laue.optimize indexing. And it's referred to as such because we take the indexing results from this initial indexing, and then we apply some polychromatic algorithms. So we can assign wavelengths to every reflection that are in the data set. And then using those wavelengths, we can also refine slightly the crystal orientation that's in there, and then assign Miller indices for all of those reflections that were not able to be indexed by the monochromatic indexer, because now we have wavelengths assigned to them. So it's easier to calculate their HKL indices. There are various parameters in this program, and this one is the first of these that's not a wrapper around dials. This is a fully Laue-DIALS program. Uh, the key required information here is you'll need to provide bounds on the wavelength spectrum. So this lam min and lam max, and then you'll need to provide a d min. And what this essentially does is internally, this program works by predicting all the feasible Miller indices that would be within the bounded area of these two wavelengths in this d min. And then does some filtering based on harmonics, what's closest to what peak wavelengths and such, to get down to what Miller indices actually show up on your detector, and assigning those to the reflections that are in your strong spots. And so you can also, at this stage, we often find during Laue.index, because it's a monochromatic algorithm, it will often assign a unit cell that's a little too small or too big to compensate for the fact that it doesn't really know the wavelength of these reflections. So you provide a wavelength that's 1.04 angstroms, for example, but the reflections might be scattering at slightly higher or lower wavelengths. And for the Laue polychromatic paradigm in general, the unit cell and the wavelength are kind of complementary values. So small adjustments in one can be compensated for adjustments in the other. And this is often the case out of Laue.index. So if you want to work with the correct unit cell that you know, Laue.optimize indexing does allow you to provide a unit cell that overrides the unit cell from Laue.indexing and then adjusts the wavelengths and scales them appropriately afterwards. But that is not necessary for this step. If you want to use the initial unit cell that you get from Laue.index, that's totally okay also. So just as a demonstration, I mentioned before that this had just under 25% straight out of Laue.index. So that these are the indexed spots out of Laue.index. And then our optimized indexing code can take this to as much as capturing 99% of the spots on an image. And this is really the key thing, the key step that allows us to do Laue indexing. Otherwise, you'd be throwing away the vast majority of your data. So this Laue.optimize indexing code, which also I should say now that we're transitioning to giving it an alias of Laue.assign, but they're the same program. That's this step, and it allows for capturing all the reflection data that you have, assigning wavelengths to all of it, getting Miller indices, so that you have enough data to do geometric refinement of your experiments. And so this essentially now you're allowed to vary things like the detector model, distance, beams, and various parts of the experiment that we held fixed in the previous portions of this pipeline because we were focused on assigning Miller indices. At this stage now, you can freely refine some of those variables. I should mention that for the unit cell, for beam wavelengths, and for the detector distance, you'll generally want to keep two of those constant because they all compensate for each other. So generally what I would suggest is keeping a unit cell axis constant, so the A-axis, for example, and then keeping constant the wavelengths or the detector distance, one of those two, whichever you feel, pretty confidently. And that allows the third then to freely refine. And I think by default, we keep the detector distance and the unit cell axis, but those can be overridden if you wish to change those two. And you can also use, there are a few utility commands. So one of them is Laue.plot wavelengths, and that just shows you all of the wavelengths that are in your reflection table. These are useful diagnostics for after geometric refinement, because ideally the reflection wavelength spectrum that you see from your data looks very similar to the actual X-ray spectrum of your source. And so, these match pretty closely to the biocar's X-ray spectrum. And this is what exactly you want to see. If this is an absurd spectrum, then you'll know that something has probably gone wrong somewhere in this pipeline. Similarly, we have tooling for computing image RMSDs, and this is for the reflection centroid data, and it's particularly useful for identifying images that, for whatever reason, we're not able to refine very well. So generally speaking, you want to have rather sub-pixel RMSDs over an image. And in this context, these pixels are 88 microns on one side, square pixels. And so if you're getting images that have several pixels for their RMSD across the image, those are generally images you're going to probably want to throw out of further processing. And that's something you can do with some scripting with reciprocal spaceship, which is another package that we have. And then we probably are going to develop some command line utility for that too. Now, once you have a refined experimental model, you can take this experimental model, put it into Laue.predict with wavelength bounds and a dmin bound like we had before. And then what this prediction code does is it takes all of the strong spot data that you have, and then using the experiment model that you refined is able to find all where hypothetically at least all the weak spots should be on your detector so that you can integrate them all appropriately. And so a little extra thing that we have here is that like Laue.optimize indexing, it does this first by finding all of the feasible HKLs that exist within the bounding boxes. So the lambda min, the lambda max, and then a d min, which is a circle out there. And in a perhaps easier to read space, we have resolution versus wavelength here. And all these green spots are the feasible predictions that lie within that range. But you'll note that the observed reflections, which are these orange spots, for the most part don't occupy that same space. And that's because of this phenomenon called resolution dependent bandwidth. And so what we did was we developed a kernel density estimator. We used it from SciPy, and we apply it to all of the strong spots in the data set that we can find to try to learn this bandwidth as a function of resolution and to build probability contour maps in this resolution versus wavelength space of how likely a reflection is to be seen on a detector as a function of these two variables. And so we use this KDE in conjunction with either default probability settings or user provided probability thresholds. And that gives us which reflections to include as predictions on an image to integrate after and which to reject as being improbable. And so you'll see that there are various contours here. These are at different probability thresholds. This is settable by the user, although I will admit that the setting is not very obvious in terms of what to set it to. But this is how that functionality works. And that then gives you all the predicted spots that you can run the Laue.integrate code. And this is just a summation integration algorithm. It finds all these spots, it builds little elliptical profiles around them, and then it just sums up the intensities in them. One caveat that I should note with this is that it is not particularly good at dealing with spot overlap. So if you have overlap spots, what this program does basically is take those two spot centroids, and then in the middle of them, it just kind of builds a line right there. And intensity on one half of that line goes to one spot, intensity on the other half goes to the other spot. So there's no kind of deconvolution of spot spectra, intensity spectra profiles over there. So we are working on developing better integrators for that. But in the absence of spot overlap, you should be able to integrate just fine. And then that will write out an integrated MTZ file, which will have this metadata inside of it. And we built this specifically so that you can pass this off to Careless, which is code that Kevin Dalton, who is in our group, also wrote. And this is merging and scaling code that handles many things, but in our case, wavelength normalization and harmonic deconvolution. That is something that's usually a problem for Laue data. We don't have to worry about that thanks to how Careless works. So you need to provide it metadata keys, an MTZ file, and just what you want to call your output files. So here's an example Careless script. At the top here, if you're working on a computing cluster, these are just some common Slurm parameters. These might vary a little bit depending on what jobs you're kind of running. This is an example of the integrated MTZ file that we would be providing straight out of Laue.integrate. These are the metadata keys, and then there's also some other arguments that you can put into Careless. So Careless has many different functionalities for this. You can change iterations. Dmin is something you'll probably want to change based on your CC halves in further processing. And there's many other functions in careless that you can do, but this is what we use for this data set. And then this is the actual careless call. So careless poly providing these base arguments, the empty Z file, and then these output file names we'll put into a directory, an output directory. We call it careless merging here earlier in the script, and then prepend all of the files with DHFR. And then for this DHFR experiment, then this is our completeness, which is generally pretty good, out to 1.5 angstroms. And our cross correlations, coefficients between half data sets are, we have these out to 1.5 angstrom resolution. And this is just a sample table that you would get out of, say, Phoenix refinement. So if you run phoenix.refine with some settings file, or if you run it through the GUI, these are R factors bin by resolution that we were able to get out of this. So what are we working on then for current development? Well, major features are pink indexer. So right now, the version that exists presently that you can download, it supports scan-based data, but it does not support serial data right now. And pink indexer, we're planning on using to allow for a serial pipeline. So pink indexer has been included. The indexing algorithm was initially added in DIALS version 3.18. There have been some slight improvements and refinements to the algorithm. So DIALS version 3.21 is the earliest I would go if you want to use this algorithm. And this pink indexer support is the thing that we're most currently actively working on providing into Laue-DIALS, and we're hoping to have that done as our next major release. We're also working on adding natively polychromatic geometry refinement. So the current refinement that we're using is kind of a hack workaround of monochromatic refinement. Essentially what we do is create an experiment model for every reflection, where everything is the same except for the beam object in it has a wavelength that is associated with the wavelength for that reflection from Laue.optimize indexing. And this was kind of a workaround dealing with monochromatic refinement algorithms that we have. One of the limitations of this is that we can't take experiment objects that would be common across a whole data set. So for example, a detector, and refine them across a whole data set. Every image has its own experiment model, its own detector, its own crystal, its own set of beams. And we would like to be able to refine a detector across the entire data set for scan-based data. And so we've been working with David McDonough on introducing some natively polychromatic geometry refinement code that builds in wavelengths into the loss function for geometric refinement. And then, yes, quick question? No? Okay. And then improved integration routines. So ideally being able to deal with things like spot overlap, and then just generally improving the quality of integrated intensities that you're inputting into scaling and merging. These are probably our three major projects that are going on as current development. We're all, of course, also open to bug fixes and like minor quality of life improvements, and I'm perfectly happy to do those. And that's something that we continually do as well. So the version that was published in the paper is right now on PyPI. You can install it right now with, if you have a Conda environment that has dials installed in it, you can just pip install Laue-DIALS and you should be good to go at that stage if you have dials and Laue-DIALS in that same environment. Yeah, so that's really all you need. You don't need to install everything else separately. It's all included with the package. If you want to work on this as a developer, you'll just need to add a little flag that downloads some additional documentation tools like Sphinx, for example, if you want to help contribute to documentation or building other portions of the code. But if you just want to be a user, this is all you need to do. Conda install dials into an environment and then pip install Laue-DIALS, and you should be good to go. And Laue-DIALS in general is part of this larger GitHub group called Reciprocal Space Station. This is where many useful softwares for crystallographic analysis exist. So Laue-DIALS is part of this, Careless is part of this, Reciprocal Spaceship, which is mostly just like a version of Python pandas applied specifically to crystallography, and other tools that our group produces will be put here too. And this is an open group. Everyone can contribute. We have a website online. We have a Slack workspace that also has development channels for these. And then also if you need help for, say, Laue-DIALS or Careless, you could also get help there because the developers are all active there, including myself. So if you need a workspace invite for that, feel free to email me or any of the other administrators of that reciprocal space station group. And then just acknowledgements. These people have all been pretty instrumental in helping to build this software and make it a reality. At Harvard, of course, I'm in Duca Hextra's group. Several grad students and some graduated now helped also with developing Laue-DIALS and applying it to data sets. Of course, we have our many DIALS collaborators at Diamond and Berkeley Lab and at SLAC. And then University of Chicago, specifically through BioCars, has been also really helpful in providing data and support for building this software. And yeah, if you guys have any questions, now's the time.